All right, last topic of our substitute video lectures after the big snowstorms of last week, enzymes. You can't take a biology class without talking about enzymes in some capacity. They are absolutely vital for life. They're one of the things that really make life possible. They're often called the workhorses of the cell, the workhorses of metabolism. Come on, my pen won't turn on for me. Here we go. Okay, so we've been talking about metabolism. You have all these chemical reactions that take place that break bonds or create new bonds, anabolic and catabolic, endergonic, exergonic. Be sure you review all that terminology. Now, so you got all these constant chemical reactions taking place inside cells. Do those chemical reactions just take place on their own? They could. If you had a protein, which is a chain of amino acids, yes, that protein, if it just sits around in the watery-based fluids of a cell, it could gradually break apart into its original set of amino acids. Or if you had two amino acids floating around in a cell, they're diffusing, moving around, they're going to bump into each other at some point and form a bond. That's possible. It could happen. But cells, living things, can't rely on random chemical reactions sustaining life, sustaining their metabolism, sustaining growth. We need something to force these molecules to happen. Like if you want two amino acids to get stuck together, we need something to grab those two amino acids, stick them together so that they force them to bond. Or if you want to build a disaccharide like sucrose, you got to find those two sugars. We need something to force those two sugars together to build that sucrose disaccharide. This is where your enzymes come in. Enzymes are said to catalyze chemical reactions. They speed them up. They increase the likelihood of them happening. They force them to happen. Enzymes, you can kind of think of these as the machinery inside your cells that force all these important chemical reactions to take place. The majority of them are themselves types of proteins. Almost all of them are. You can recognize enzymes usually because their names tend to end with the suffix ase, ACE. Some examples: sucrase, lipase, DNA polymerase. What do you think sucrase might do? Sucrase is an enzyme that breaks apart sucrose. Sucrose is a disaccharide, so the enzyme sucrase takes sucrose and splits it apart into two monosaccharides. What's a lipase going to do? A lipase is going to break apart a lipid. Now, not all enzymes. Some students early on when they're studying enzymes think all enzymes break things apart. They do catabolic reactions. That's not true. You also need enzymes for your building reactions as well. DNA polymerase, for example, is an enzyme that builds DNA polymers. So it takes DNA nucleotides, the building blocks of DNA, and bonds them together. The enzyme that does that is called DNA polymerase. Another feature of enzymes is they can be reused. So one enzyme, say one sucrose enzyme, splits apart a sucrose molecule. Okay, then it can go off and split apart another sucrose. It can go off and split apart another sucrose. So they can be used over and over again until eventually they get damaged and wear out and have to be replaced. Okay, this is a diagram from your textbook which is showing you the components of a typical enzyme in a cell. Now, we just said enzymes are typically proteins, but many enzymes have some other components as well. The big purple blob you see on here, that's the protein portion. That's sometimes called the apo enzyme. But then some enzymes will have cofactors and or coenzymes. Okay, so like a cofactor over here, a lot of times those are metal ions, like magnesium, zinc, copper, iron, things like that. Some enzymes need those and some enzymes don't. If they need them, they've got to have those to be able to perform their chemical reaction. Some enzymes need coenzymes. Coenzymes are smaller organic molecules, usually vitamin derivatives, that are also necessary for that enzyme to be able to carry out whatever function it may have. Another important aspect of an enzyme is called its catalytic site or active site. That's another name. So enzymes are going to have some sort of particular three-dimensional structure. There's an enzyme. And then the enzyme is going to bind something to perform a chemical reaction. So it's got to have a little pocket over here that whatever that enzyme is going to act on can fit very specifically inside that pocket. So that pocket is called the catalytic site or the active site of the enzyme. Different enzymes, that catalytic site is going to have a different three-dimensional shape. 3D shape is very important for enzymes. And if you relate that back to what we were talking about with temperature requirements for growth of microbes, remember we said at high temperatures, proteins that have a nice 3D shape, they tend to denature, they unravel. If an enzyme unravels, it can no longer carry out its chemical reaction. Well, this is why you tend to die at temperatures that are above your maximum growth temperature, because your enzymes lose all their 3D shape. If your enzymes aren't working, you're going to croak pretty quickly. All right, another very important concept with enzymes, substrates and products. Enzymes carry out a chemical reaction such as A is going to be split apart into B plus C, or A plus B will be bonded together to make actually a better way to write this. You got AB and you're going to split it apart into A plus B. Or you've got A plus B and those are going to be bonded together to make AB. Okay. Again, inside cells have enzymes to catalyze or speed up or make these reactions, force them to happen. All right, so whatever an enzyme, like they're showing you over here, whatever an enzyme binds to, the input, whatever that's going to be, is called the substrate or the substrates. All right, so you see how our little purple enzyme here bound this substrate that kind of looks like a duck bill or maybe the nipple on a bottle, and that has fit inside this little pocket right here is called the catalytic or the active site. Notice that substrate fits perfectly in there. So this enzyme is tailored to react with this particular substrate, whatever that is. This other chemical molecule over here does not fit. It's not a good fit for this particular enzyme. This forms a complex between the enzyme and the substrate. And then whatever the enzyme makes or produces, that's going to be called the products. That's what gets released from the enzyme. All right, so in our little chemical reaction statements over here, AB would be your substrate. All right, and then an enzyme is going to bind AB and split it apart into A plus B. These are called your products of the enzyme. Or if you have an enzyme that takes A plus B, that's what it captures. I'm going to capture A and B and bond them together. A and B are still called your substrates. And then AB that got bonded together would be the product. So it depends on the type of chemical reaction you're doing. Is it catabolic or is it anabolic? Are you building something or are you breaking something down? But your inputs are always the substrate or the substrates. The outputs over on the right-hand side of your chemical reaction will be the product or the products of an enzyme. Here's a diagram that's showing you this with an actual organic molecule, and no, you're not going to have to draw chemical structures, but this is just showing you what's going on with an actual biologically important molecule. So you've got a type of sugar molecule here called fructose 1,6-bisphosphate. This is an enzyme that takes that molecule and splits it into two smaller molecules. Okay, so this is our substrate, this bigger molecule. The enzyme binds it, so this fits down into the catalytic site or the active site. The enzyme forces this molecule to split apart. And these are your two products. That bigger molecule got split apart into two smaller molecules. So those two smaller ones there are going to be your products. Notice the products get released, so your enzyme can now be reused. It's going to float around in the cell and find another one of those to split apart. All right, enzymes can be affected by various factors. And again, that's generally because the main part of most enzymes is a protein, the apoenzyme part. And for proteins to work, you got to have a particular 3D shape. If temperature is off, if pH is off, pH is another thing that can influence whether an enzyme works, that protein is going to become denatured. It's going to lose its original 3D shape and it's not going to work anymore. So if you look at these graphs over here, it's showing you for a typical generic enzyme, its activity, and its activity is referring to how quickly, how efficient is it at making its products? How efficient is it at binding its substrates and making its products, whatever those might be. So if you notice over here, enzymes tend to have an optimal temperature. They have an optimal pH, and if you go lower, they tend to slow down. They don't make products as quickly. If you go higher, you tend to have a pretty fast drop-off. And the reason you're having that fast drop-off is because those protein enzymes become denatured. They lose their 3D shape. You don't have your 3D shape, you can't attach to your substrates and change them. Some other things can affect how well enzymes work also. Concentration of the substrate, that's just kind of common sense. If you have less substrate available, the enzyme's not going to be able to make as much of its product. If you have tons of substrate around, the enzyme can get what's called saturated, where there's only so much product it can crank out. You only have so many copies of this particular enzyme to find these substrates and crank out product. So enzymes have their limits in terms of how much product they can crank out. Concentration of the enzyme available inside a cell, your textbook touches on this topic a little bit as well. Cells have to make their enzymes. They don't absorb those from their surroundings. And so cells are only going to be able to crank out a certain amount of these enzymes. And so the amount of product you can make with these enzymes, well, it's going to be limited by how many of those enzymes you have around, just like it's going to be limited by the amount of substrate you have around. All right, by now I'm sure you realize how vital enzymes are for cells. So the activity of enzymes, cells are pretty good at controlling the activity of enzymes, keeping them under control, using them only when needed. Because it would be wasteful for cells to, say, make too much of some particular polysaccharide if they don't actually need it. If you're a bacterial cell, you don't want to manufacture a bunch of peptidoglycan cell wall stuff that you don't need if you've already got an intact cell wall. So cells are really good at keeping their enzymes under control. A couple of ways they can do this. One is through competitive inhibition. Another one is through non-competitive. And competitive, non-competitive means competing for the active site, the little 3D pocket on the enzyme that binds to the substrate. So in purple here, that's your normal substrate. So like if this enzyme is sucrase that breaks down sucrose, the substrate there is going to be the sucrose sugar molecule. All right, so a competitive inhibitor, this is something else that's present in the cell, some other molecule that will compete with whatever normally binds the enzyme and can fit inside that pocket. And so like they're showing you over here, if the competitor is in there, the enzyme can no longer make its products. If the competitor is not around, the enzyme can still make its products, whatever those may be. That's competitive inhibition. The inhibitor actually fits into the same active site or catalytic site. Non-competitive inhibition over here on the right-hand side. In some cases, this thing that kind of looks like a yellow Hershey's Kiss over here, I must be hungry. Notice that it is attaching to the enzyme at a different location, not in the active site. But by doing that, it's changing the shape of the enzyme a little bit. If you notice, if you look closely, the active site there has changed shape. And that active site can no longer bind to this substrate, whatever it is the enzyme is supposed to change. Okay, so that's another way of blocking the activity of enzymes. If you don't have this non-competitive inhibitor around, your reaction can proceed normally. The enzyme's going to do its normal job and it's going to make its normal product. Notice on here what they're showing you is that the product of the enzyme in some cases can actually be a non-competitive inhibitor. And that might seem kind of strange at first, but remember when you take A&P, you learn about negative feedback processes. And a lot of these negative feedback processes you're learning about when you go into when you look at them at a detailed level, this is what's going on. It's all about shutting down enzymes or allowing the enzymes to carry out the chemical reactions. That's what's actually happening down at a more detailed level. So what this is showing you is cells don't want to be wasteful. So if you've already made enough of this product, whatever it might be some sort of polysaccharide or a disaccharide or whatever, that product can actually inhibit the formation of more of itself. So that way it's telling the cell, hey, we've got enough of me. Don't keep making more of me because that would be wasteful. Enzyme inhibition is also important. A lot of the drugs that we take, not just for infections, but many of the other types of drugs that we take to try to improve our health, work by inhibiting enzymes, either competing or through non-competitive inhibition. Many of the drugs that we take. For example, I've already talked about in class how penicillin and many of its relatives work by blocking bacteria from being able to build their peptidoglycan cell walls. The reason is those drug molecules inhibit the enzymes in the bacteria that build peptidoglycan. So if their enzymes can't build peptidoglycan, that bacterial cell is not going to be able to manufacture its cell walls. This diagram is showing you that now typically in like when you take anatomy and physiology, you learn that a lot of things occur in step-by-step processes. And that's including metabolism, catabolism, and anabolism. Whenever you're making some sort of a product or you're breaking something down, it's not usually just a one enzyme leads to the product type thing. Usually it's a step-by-step process that involves a series of enzymes that take place, and these reactions take place in multiple steps. That's what this diagram is showing you. Here you've got some sort of a substrate that's going to be used to make a product. Okay, so this little blue blob here is supposed to be enzyme number 1. It binds this substrate, changes it in some way. It's altered that molecule, and that gives you intermediate molecule A. Intermediate molecule A binds to enzyme number 2. And enzyme number 2 alters it in some way, makes intermediate B. Intermediate B then binds to enzyme number 3, and finally enzyme number 3 is the one that produces the final product, whatever this might be, some kind of molecule that the cell needs. Unfortunately on this diagram, I wish they had made the enzymes look a little bit, maybe they were different colors to emphasize that you've got 3 different kinds of enzymes that are binding to the 3 different molecules there in the diagram. Notice on this chart, finally, this end product over here, whatever that is, goes up here, and it does a non-competitive inhibition of enzyme number 1, the first enzyme in the pathway. So it attaches to it, changes the 3D shape of your active site right in there, And then this original substrate up here at the top can't bind to enzyme number 1. So that's going to shut down this reaction, which is going to shut down this one, which shuts down this one. And that shuts down the formation of any more end product. And again, the idea there is if you have enough of this around, then you need to shut down this pathway. Okay, and the most efficient way to shut down the pathway is to come up here and inhibit the very first step, because you don't want to be wasteful and still have enzyme 2 doing its thing and enzyme number 3 doing its thing. Or I'm sorry, you don't want enzymes 1 and 2 doing their chemical reactions when you don't need any more of that end product, because that would be very wasteful for a cell. This is an example of what's called feedback inhibition, very similar to what you guys learn about when you take anatomy and physiology. You hear a lot about feedback inhibition, especially when you study hormones. And again, why is this a good thing for anabolism and catabolism? It just makes the cell more efficient. Don't waste resources and energy building things and breaking things down unless you really need to do so. All right, as I mentioned, many of the drugs that we take artificially to try to control body processes, to control disease, to kill microorganisms inside the body, work by inhibiting one or more enzymes. All right, so I know you guys are tired of video lectures, substitutes for our snowy week last week. That's where I'm going to stop. We are going to pick up here, now that we have some background on enzymes and an introduction to metabolism. We're going to talk more about how microorganisms perform their catabolism, their catabolic reactions, to harness energy and make the ATP fuel that they need to power all of the things that the cell needs to do that requires work. So this is where we'll pick up when we finally get back to class next Thursday.